dobry wszystkim, którzy już są przed telewizorami. Ja przejdę na język angielski, ponieważ gościem naszym jest Shawn, łącznie bezpośrednio ze Stanów Zjednoczonych. Od niego jest godzina 3 nad ranem, więc, ale wygląda bardzo dobrze, to znaczy nie jest zaspany. I'm switching to English now. Okay? Welcome everybody. I just introduced the situation that we have a connection with the USA. That was actually presented in the invitations, so I think nobody is surprised. We are meeting today in Life Science Discoveries. Discoveries Life Science, Poland, in Poland, in Kraków, sitting in Life Science Park at this moment with Tetyana, and also we have a guest from the same company, Shawn Fahl. Shawn Fahl, connecting with us from
Huntsville, Alabama.
Alabama. Okay, never been.
Yeah, it's Huntsville, Alabama, where last year, Shawn, Discovery opened most innovative, very beautiful, big, attractive business center, which looks awesome. And really now, probably this is one of the biggest, it's kind of the Silicon Valley, but even more cool. So this is a biotech valley in Huntsville, in Alabama, in this new building, putting together different departments. Shawn today will explain and show how the global DLS looks like, because in Poland we are working probably, everybody know us in the biospecimen directions, but today global Discovery Life Sciences is much, much more than only biospecimens. And this is kind of the full service company with this very beautiful office in Huntsville. And every time I'm mentioning our headquarters in Huntsville, I have this association about similarity of life science cluster in Kraków and the history of Discovery Life Sciences in Huntsville, where everything started from the cooperation with the Hudson Alpha Institute. The concept at the beginning was the same, to create support full services for science to move forward quicker. So, Shawn, the floor is yours.
Awesome. Thank you very much.
Just a few words more. We'll be talking today about personalized medicine. So it's actually very, very important subject for our community, because this personalized medicine is something we have on the top of our minds since many years, and we believe it will be strongly developing now. So it's the right time to have you presenting this discoveries from life sciences.
Fantastic. Thank you. Yes, personalized medicine is the future, and I'm so excited to be on top of that. Okay, so let me go ahead and share my screen here. Okay. Okay, so thank you everyone for joining, and good morning. So today I'm going to talk about what we've been doing at Discovery, really to help hopefully shorten the window required to move a new drug target from early discovery to clinical reality. So for Discovery, we are based in the US and Europe, so sites across both the West and East Coast of the United States, as well as multiple sites within Europe. And really this gives us a nice global footprint. So these sites do have specialties in what they do, but multiple sites will usually have multiple different business divisions in them. Is really key, because one of the big things that we strive for is really speed, is being able to move a sample or a study through multiple different formats as quick as possible. So Discovery is basically about 6 to 7 different divisions. They are roughly split into the left side, which is a products division. This is a division that the end user will receive a physical specimen to work with. versus the right side, which is our services division. In this case, we are delivering the actual data the researcher needs for their study. So in the products division, there are 3 different divisions. The far left is biospecimens division. So this is where we will spend most of our time today. And in our biospecimens division, we really focus on 3 areas: tissue, blood and bone marrow, and then other biofluids. And in the next slide, we'll go over a little bit more about what those are. We then have a cell and gene therapy arm, which is also known as all cells. So this focuses specifically within the cell and gene therapy field, providing leukopaks, so large-scale blood collections, to those manufacturers who are working either in stem cell technologies or CAR technologies. The third products division is our preclinical division. So this is focusing really on drug toxicity. So they focus a lot on liver derivatives, things like hepatocytes and microsomes, and really understanding if any drug compound that's in development is just inherently toxic. In our services division, we'll start actually on the right and work our way over. So our molecular pathology division, this focuses really heavily on late stage and early stage clinical trials. They're taking companion diagnostics and patient enrollment using primarily IHC, although there is some additional offerings within this division. Our proteome center, this focuses on all proteomics-based technologies, so things like mass spec, Olink, Luminex, really trying to understand at more of an unbiased and high level what the proteome looks like. And then our discovery genomics division. And so this is really the division that is all of our genomic sequencing, so both on the RNA and the DNA side. And then finally, in the middle here, and this is where we'll spend a lot of time today as well, is our cell biology services. So these are our services where really viable living cells are crucial. And so in this case, we do a lot of things like flow cytometry, single cell genomics analysis to really understand at the single cell level what's happening, especially as we move forward in treatments. So for our biospecimen formats, there are really 3 large groups. So we have access through our global clinical network. So we do manage a network of hospitals and practices both in the United States and in Europe. And from these, we can get various biospecimens. So the most prevalent is our blood biospecimens. And so we do receive these as whole blood and then in our processing facilities. So we do have one here in Huntsville and one in Sofia, Bulgaria. This blood will be broken down into plasma sera, double spun plasma, Buffy coat PBMCs. This happens not only at our sites, but also at our partner sites. And so it gives us a really wide range of specimens to offer. The second group is our tissue-based group. So this includes both bone marrow and solid tissue. Excuse me while I get the cat out of the way. And so for bone marrow, this primarily is in the form of a cryopreserved BMMC fraction, whereas in tissue, we have access to flash frozen tissue, so frozen immediately at the site, so no longer viable, but of high quality, fixed FFPE tissue, which is the most prevalent. And so we do have a rather large, robust inventory of this, and then DTCs, which is a little more of a unique sample type, which we'll discuss in the next slide. After that, it's really everything else. So things like swabs, urine, sputum, additional biospecimens that could come from patients. So really, we started DTCs probably about 10 years ago now, because we used to provide fresh tissue from our sites to our end users. The issues with fresh tissue can be getting it from that site to that end user in a time frame that it can be used. And then there's some additional issues around subtypes and composition and things that we'll talk about in the next slide. But what we decided was, with this tissue, by the time it got there, usually wasn't usable. So what we did instead was brought it to our facilities. In our facilities, we go ahead and disaggregate that solid tissue and freeze it down into vials. And so what we've done is we've actually taken the entire tumor microenvironment and kept it largely present. And this means it doesn't matter what part of the tumor that the researcher is interested in, it should be present within this frozen vial. And what this allows us to do is get a very robust repository where we have hundreds of samples from hundreds of patients, so that when we create cohorts, we can create large cohorts quickly. These are usually characterized and with usually the most prevalent biomarkers that are clinically relevant. And then we also have normal tissues as well, which really serve as controls for a lot of these studies. So we did have a lot of work up front with these dissociated tumors, largely to show that they can still function and work as though they were fresh tissue. So they can undergo DNA and RNA sequencing. We can use them for flow cytometry to look at the protein expression. These do grow in culture. So these are short-term tumor spheres where the tumors will make these spheres in culture, or long-term organoids where they basically grow as a cell line. And then they can be functional. So they can represent everything that the parent tissue did. And so really our focus has been looking at the drug pipeline, and the idea that the drug pipeline can take 10 or more years, a lot of money, and ultimately potentially not be successful. And really that you've got a lot of work going on early on to decide, is the drug target I'm looking at present? At what level is it present? Doing your drug discovery, your preclinical, all through clinical trials. So at Discovery, what we're trying to do is, by putting basically all of the different technologies and samples required for this within one facility, within one company, we're really trying to shorten that time frame, ultimately reducing costs. And so really how we've tried to do this is twofold. One is just having a large, robust biobank. So this is a lot of information on this table, but basically what we're looking at here is, we said, okay, if we look at about a dozen different cancer indications, including heme cancers, so multiple myeloma or AML or solid tissues, in the network that we manage, how many unique samples could we get in a time frame? And so for some indications, colorectal cancer, in a couple months, we could have 30 to 40 unique cases, which is a lot, which is really, really good. But it still has some caveats in terms of, if you're looking for rarer indications, melanoma, you can only really see 5 within a 4-month time span. And it becomes harder and harder to get these samples to the right end users in enough quantity to draw conclusions. By having the biobank, what was really in the single digits or 10s or 20s really becomes the hundreds. And so now if a multiple myeloma researcher wants to come and do a study, they could pull from the bank up to 700 unique patients, or for solid tissues up to 100 different ovarian cancer patients. And so again, we're trying to shrink that timeline down by giving more samples quicker, so that really questions can be answered faster. And so again, what we're trying to do is really move from this idea that, and this is really, it says current medicine and future medicine. I would argue that we're more on the future medicine side, but that when a patient comes in with a specific disease, I think in this graph we're talking about colon cancer, they receive the same treatment. And some patients might have a great effect, some might have no effect, and some might have adverse effects. But what we're moving towards is where that same group of patients comes in. We take samples from those patients and understand what mutations do they have, what biomarkers are they expressing. And each group then gets a medicine personalized to them, such that instead of having either no effects or adverse effects, everybody is having a positive effect to their treatment. And so really the first step in this is taking that biobank and really making sure that it has the most information for end use. Because when the idea of personalized medicine is, you have to be very targeted with the group that you're studying, because not every patient will respond. And so if we look at, again, either the fresh component or what might also be in some other large biobanks, is we don't know a lot of the really big factors that are required for these decisions, such as the patient demographics, the diagnosis of disease. A lot of times it's suspected but not confirmed, subtype and stage of the disease. And then specifically for things like personalized medicine, is the cell type I'm targeting even present? Is the biomarker I'm interested even present? And so with a fresh tissue collection, that becomes really difficult. And so what we've done is made sure that any tissue that comes in, we have a base set of data that we get from the site, the clinical site. And that includes all that patient demographic information, which comes on a case report form to us. And then we actually receive the pathology reports from our sites, which means that we can actually confirm the diagnosis, confirm the subtype and stage, so that we know exactly what this tissue was. For the cellular composition, this has been a focus of ours pretty much since the beginning, because the idea that we have a tumor, and while there are a lot of tumor cells in there, there are a lot of other cell types that are really crucial for that tumor to grow, including fibroblasts for support, blood vessels to bring blood and nutrients to that tumor, as well as a pretty substantial portion of the immune system. And that's been a lot of focus, really, is how to impact that immune system to start recognizing and potentially treating that tumor as though it was an infection. And that's the case study that we'll go through today. So what we've done is every tumor that comes through our lab, we profile it to know exactly what cells are in there. And so in this case, we use flow cytometry. The cells are labeled with fluorescent antibodies to let us know what type of cell they are. And then we hit them with a laser, and that lets us know the percentage of each of these cells. And so in this case, we can see this box up here actually represents the tumor component from this tumor. And while the bottom right here shows all the immune cells, and then this is the very immune cell subsets. And so again, in the immuno-oncology world, using the immune system to attack the tumor, this is really crucial for researchers to understand if their actual immune cell is even there. So now, in addition to having all of the information on the patient, the pathology information on the tumor, we now know the exact composition of the tumor at the cellular level. And then really the last part was a lot of therapies now are predicated on knowing the clinical relevant biomarkers. And some of these we do receive from the sites, and others we go ahead and test in-house. And so this can include things like PD-L1 expression. So PD-L1 is going to be the focus of the case study, but it is a cell surface receptor that will turn off the immune system, which is obviously not what we're striving to do a lot of times in immuno-oncology. We have microsatellite instability, which is really a readout of genomic instability. Again, this will be part of the case study for today. HER2, ER, and PR. So these are the three receptors really for breast cancer predominantly, and things like triple negative breast cancer, in which all three are lost. As well as HPV subtyping, which can be a driving force for things like head and neck cancer and cervical cancer. So in addition to knowing the cells, we actually profile our tumors for these factors as well, such that now we've not only got information from the clinic, information on what cells are in the tumor, but then also know all the clinical biomarkers as well. And so that does lead us into the case study. So how are we using all of this data internally to demonstrate or identify new findings? And so again, the idea of PD-L1 is that it is present on the surface of tumors. Not all tumors, but it is present on the surface of some tumors, particular things like lung cancer. T cells in the tumor actually express a molecule called PD-1. And when these two interact, it effectively turns that T cell off. So this T cell is a major component of your immune system that likes to clear infections and clear infected cells. And so by being turned off, even if this T cell could see this tumor cell and project it and potentially kill it, it can't because it's turned off. And so there are a number of therapies on the market now which will actually interrupt this interaction. By preventing this interaction, this T cell now sees this tumor cell as danger. It sees it pretty much like an infected cell and will go ahead and kill it. And so this is a survival graph from one of the very early studies. So in this case, we're looking at the survival of patients on the study, either receiving the standard of care, which is usually some form of chemotherapy in the red, or receiving a therapy, in this case it's pembrolizumab, which interacts and interferes with that PD-L1, PD-1 interaction. And there was a clear survival advantage of the patients that received this immunotherapy, that received the inhibitor, and really shows how great these immuno-oncology drugs work. but there's still work to do. So there's still obviously a subset of patients that didn't respond, especially early on. We did see still some reduced survival throughout the length of the study. And so really there's additional questions, not only in how these drugs work, but how do we identify patients better to make sure that those patients that will have the best chance of survival are receiving these therapies. So this therapy, I really like to highlight this slide because it really was first approved by the FDA in the United States for melanoma back in 2014, so only about a decade ago. And since then, it keeps getting approved for new indications, honestly, yearly. We stopped this graph at 2020, but even still today, there are still new indications and new cancer subtypes that this is being approved for. The really interesting thing, and really where we talk about personalized medicine, is as this therapy was coming and being approved, there were clinical tests also approved that could stratify patients as to whether or not they should go on this therapy or not. And so the first one was an IHC test for actually even the presence of PD-L1 itself. If the tumor doesn't express it, it would be highly unlikely that the patients might respond to the treatment. And excitingly, this companion diagnostic was developed at Discovery Life Sciences through one of our groups that was known as Qualtech. And this is still used in the clinic every day to stratify patients in these various indications as to whether or not they should go on this therapy. The other two are interesting, which are MSI, so microsatellite instability, and tumor mutational burden. And these were very interesting approvals because this was the first time in which a diagnostic was approved that was independent of the actual indication. So MSI and TMB aren't required for a patient necessarily to come in with lung cancer. They can come in with a number of different indications and be tested for these and potentially go on the therapy. So it was really a groundbreaking approval because of how broad it was. So again, for MSI and TMB, the idea here
Shawn,
is... Oh, yes.
Can I have a question?
Of course.
At this moment, but the question is about a few slides back, but you don't have to go back. Okay. It's you are presenting your kind of experience and also the approach you have in the company to collect information regarding which is related to different types of diseases, etc. in personalized medicine. I'm not specialist, so my question may be a little bit strange, but I started to think. You collected a lot of information about let's say human being and human organism. The question is how much, what percentages, if we can think about that at all, what percentage of this information you collected now represents of the total knowledge? If we wanted to know everything about human being, is it 1% or 50%?
Right. That is an amazing question. So we have in the database that information on thousands and thousands and thousands of individuals. It's a great question because it's something we've discussed quite a bit, which at this point we largely provide the specimen with that data and then services on the specimen, but we actually have so much data that it actually begs the question of how do we leverage just the sheer amount of data that can be evaluated, can be data mined to discover new trends that we haven't seen before simply because we have so much of it. Did that answer the question?
Not necessarily.
Okay.
That's why I said it would be strange. My question was rather, I just imagined that sooner or later we will have knowledge about 100% what's necessary to know about person. Yeah. In terms of molecular biology, etc. And question is how far we are from having 100% total knowledge. Yes.
I think we're closer than we know. So with a lot of these technologies that we're using now are so deep in that we've moved from where we're testing for a single marker or a handful of targets to testing thousands of targets at one time. And so I think, exactly as your point, I think that we're getting very close to where we'll know everything about a patient when they come in just by doing some very simple tests. And I think that's way quicker and coming way faster than we all realize with the speed at which these two new technologies that can do those type of analyses are coming online.
Thank you.
Yep. So for MSI and TMB, really what we're looking at here is the idea that they are readouts of genomic instability. So one of the characteristics of tumors is a lot of mutated DNA. And what mutated DNA is essentially DNA that your body would not have recognized as you. So it almost appears as a virus, as a danger signal. And so in this case, that mutated DNA can be recognized by T cells and other cells of the immune system in the tumor and elicit that response. And so basically we're using things like MSI and TMB to do a readout of that. And so we focus largely on microsatellite instability because it's a very easy readout, very quick and very, very highly standardized. And so really what we decided to ask was a question of, can we start taking some of these different biomarker tests and start putting them together, start to see if there are trends. Okay, so now we have data on patients, on samples where we know their PD-L1 status, we know their MSI status. Is there any sort of correlations there that we can draw, or can we use that information to ask new more unbiased questions? So PD-L1 has historically been looked at by IHC. So these are some slides that were performed looking at PD-L1 expression, which you can see is this dark brown color. And so in this lung cancer case, our pathologist reviewed this and said 100% of this tumor is expressing PD-L1. In this lung cancer case, very little, if any, brown staining was observed. So this lung cancer, therefore, is devoid of PD-L1. And then this kidney cancer sample was somewhere in the middle. And so for these samples and for a handful more, we not only had this parent material, but we also had that dissociated tumor cell fraction, which gives us the ability to do a lot more different assays with it to answer kind of these more exploratory questions. And so using these same tumors, what we're looking at here is flow cytometry data. So in this case, we are looking at PD-L1 expression in this red histogram here. And we use a control just to understand what the background levels. Basically, if the flow cytometry can have a lot of background fluorescence, and we want to make sure if we see a signal that it's a true signal. And so for the tumor that the pathologist reviewed at 100% PD-L1 expressing, we see a shift. So this histogram, the red histogram, moves away from our background. In the case of 0, they can perfectly overlay. And then in the case of the 28% tumor, we saw this slight shoulder, which if you actually ask the software how many or what percentage of the cells were in the shoulder, it came out to be very, very close to 28%. And so using this over a handful of tissues, there was a high degree of correlation between the PD-L1 IHC test, which is the clinical test for PD-L1, and what we could do by flow cytometry. And so now we have the ability to use flow, which can do tens and 20s of different markers at one time versus IHC, which is largely restricted to one or a handful. So again, we can get a little more granular with what we're analyzing. On top of that, we overlaid MSI status. So MSI status is basically two buckets. MSI high, so these MSI high patients, which are in the purple, are those that do have high degrees of genomic instability. So would be predicted to have new mutated DNA that the immune system would respond to. Versus orange, these are the stable patients. So these patients show no evidence of genomic instability, at least at these specific assay, therefore not necessarily predicted to have those new tumor antigens, at least at a level that would be worth modulating the immune system. There are three tumors types that these are most prevalent in: colorectal cancer, gastric cancer, and endometrial cancer. And so we surveyed a number of our dissociated tumor cells. So similar to PD-L1, using these MSI tests and saw that anywhere from about 20 to 50% of these tumors were MSI high. And so now we're going to ask just basically a simple question, which is taking our MSI characterized cells along with our flow verified assay for PD-L1, is there any correlation between the MSI status and the receptor expression of either PD-L1, which again is the marker on tumors that is going to turn the T cells off. It has a partner receptor known as PD-L2, which has a similar function, or PD-1 itself on the presence or on the surface of a tissue or a tumor. And so that's what we did in this study. So this is a lot of information. So I'll spend just a touch of time going through this. So each set of bar graphs is a different type of cells. So either your CD4 T cells or your CD8 T cells. Your CD8 T cells are the ones that are killing cells, while your CD4 T cells are ones that are basically getting the immune system functioning. So we want to know, is there any correlation there, as well as the tumor cells themselves. In each group of three, we're looking at it for indication, either colorectal cancer, gastric cancer, or endometrial cancer. And then the orange, again, represents the MSI highs, while the purple represents the MS stables. And so what we saw by doing this experiment is that irregardless of the MSI status, PD-1 expression, so that inhibitory receptor that turns off the immune system, was very high in CD4 and CD8 T cells, which is fairly well known out there. But these cells really don't express PD-L1 or PD-L2, the ligand required to interact to turn the cell off. Now, interestingly, in these three tumor types, we didn't see much expression of PD-L1 or PD-L2, regardless of MSI status. Little surprising, although there is research out there that these don't express a lot of it on the surface. And we know that if we put a lung cancer sample through the same study, that we see robust and a lot of PD-L1 expression. So in this case, even though we were trying to see if there was a correlation, it appears that there's not. And even more specifically that, at least for these samples and for many more that we've tested that aren't on this slide, they're really not expressing these markers on their surface. So while this was very targeted, and kind of going back to the question, we wanted to know broadly, like, how can we know overall, what is this tumor expressing? Because there's a lot of insight that can be done if we looked at this unbiasedly, instead of looking at a handful of markers or looking at thousands of markers. And so is there more of an unbiased approach that we can use to uncover novel targets? And what we've moved towards is this idea of single cell transcriptomics. And this is really exciting because if we think of the tumor again is a ton of different cell types, all that have functions to help this tumor grow. When we do bulk RNA analysis, we're taking that entire tissue and getting the RNA of all of those cells together. And so when you read that out, what you get is basically the presence of different genes, but at roughly an average from all of those cells, such that if your gene of interest or what we're trying to study is, let's say in this red bar, it would appear that, it wasn't very prevalent in this tumor. For single cell analysis, what we do instead is we actually sort out the individual cells that made out this tumor, and we actually look at the gene expression specifically within that cell. And why that's important is now because we are going to personalized medicine where our therapy is going to affect specifically T cells, we want to know what those T cells express without having a tumor cell or a fibroblast signal that interferes. And so in this case, now we see that red gene that we're mostly interested in, that's restricted to this yellow cell population. And maybe that's the cell population I want to target. And in that case, this looks like this could be a good therapy. It is expressed, and it's expressed where I want it to be. And so that's really where we've been moving towards, is these idea of single cell transcriptomics. We also do some proteomics here, where we can look at, instead of tens of markers, thousands of markers at the single cell level. And so this is what that data looks like. So this is actually a colorectal cancer sample. Every dot that you see here is an individual cell. And so at each one of these dots, we know thousands of different genes that are expressed. And what we can do is say, okay, we know that T cells and only T cells will express gene X. And so we can go through and say, okay, so all of these cells up here express gene X, they're all our T cells. And then, further define those as, they express the markers of CD8 T cells or CD4 T cells or T regulatory cells, which is a cell type that, again, helps suppress the immune system. And so we can really get granular as to what these express, but we can also unbiasedly look at any gene really in the human genome and understand its expression pattern within the tumor fraction or the immune fraction, just to make sure, especially if we're looking at new targets, that they're not expressed in cells we don't want them to be. We have a protein that's expressed in tumors, but it's also expressed in blood vessels. So giving that to a patient might not be great because it's also going to affect those cells as well. And so what we've done now is taking tumors that we have identified the MSI status, so either MSI high or MS stable, and basically run these single cell analyses and then asked what genes are expressed within the tumor specifically compared to the immune fraction. And so what this gives us is about the top 50 to 100 genes that are different. So these genes in red, so each one of these little bars represents a unique gene. So these are the genes most highly expressed in tumors, not expressed in the immune cells, in the inverse. And what we can now do is say, in these two cohorts, are there genes that are shared? Genes that are on or present in MSI high tumors, a lot of genomic instability, but not present in MS stable tumors. And so when we did this initial analysis, 2 cell surface receptors, which is good because those are easy drug targets, were identified, CEACAM5 and CEACAM6. And this is actually even more exciting because we did this not going in looking for these markers, and there is a lot of research out there on CEACAM5 and its role in cancer. And so again, it shows how these data sets can actually be used to prove already established hypotheses and new biomarkers or drug targets on the market. But what was really interesting about these 2 markers is that when we looked at the percentage of specimens that had those expressed, in the MS stables, it was almost all of them expressed CEACAM5 and about half expressed CEACAM6, where they were very rare in the MSI high fraction compared to the stable fraction. So already we can just, with limited data sets, start understanding the dynamic relationship between all the genes and these various already clinically approved biomarkers to really start stratifying patients even more. And again, kind of the overarching, what we're really looking to do here is to shorten this pipeline. We want to give as much data up front when we're doing initial drug discovery, initial biomarker or drug target identification, such that researchers aren't spending time on drugs or on targets that with just data mining we would know, or with just having highly characterized biospecimens and testing them up front, we would know they weren't good targets, to really reduce this timeline down immensely as much as possible, especially in this preclinical window, such that we can move into the trials faster, getting these approvals faster, getting these therapies to patients faster, and that way they will receive the most benefit. So that's the talk for today and what we're doing at Discovery across the board, from our biospecimens to our services. Happy to take any questions, any comments, anything anybody would like to discuss. So thank you.
Thank you very much. If someone has questions, please ask. Please don't be shy. Ask questions. Otherwise, I will ask, and we can have some discussion. But first of all, congratulations. I was afraid that you have 29 slides and you will not make it in 1 hour. You did it in half. So you are a great presenter. Monika Ślęzak. You have to unmute yourself, I believe, then ask. We don't hear, so if this is technical problem, so you try to solve them, and I would maybe ask a question. Grzegorz, do you see?
No, sorry, I can't. Maybe you can write your question on the chat.
My question is, what do you think, what will be the next bold discovery, life science discovery, not discovery life sciences, but life science discovery regarding the personalized medicine? What we can expect from the science and from the innovators?
It's a great question. It's an amazing question. I think really what we're looking at now and the next thing in personalized medicine is, a lot of what we're doing right now, especially with these, these immunotherapies, is turning the immune system back on. The immune system itself is insanely complex. And so I think that's really where we're striving towards, and there's a lot of research towards, is the T cells themselves. What are they recognizing on the tumor? So we're basically in the idea of PD-L1 inhibitors, is we're basically just stopping that interaction and letting them do what they need to do. But we don't know exactly what they're targeting and how they're targeting it. I think that's really the next breakthrough in personalized medicine, is where you come in and your immune system is profiled. And okay, these T cells that you have, we know they're towards a target on colorectal cancer. We want to get those T cells to function better. And I think that's really where we're going towards, is understanding that immune system at the most granular level, such that you can understand what the T cells are looking at and how they're going to respond in a very specific manner.
I think I solved my problem. Can you hear me now?
Yes.
Yes, perfect. Thank you very much for the interesting presentation. And I was wondering, because you make a huge amount of work, I was wondering if you're also doing some work in the partnerships or within the European projects. If so, what kind of partners you're looking for your collaboration and for the further development of this platform?
It's a great question. So we're always looking for partners. We partner here with various. I'm in the United States, so we partner with various entities here to do studies. Largely, what that is just have an idea, come talk to us. We're always happy to help out. We've got great teams over with what Tetyana's doing over there. Great teams to discuss new projects with, and just amazing people to talk to. So any projects, definitely let us know. We love to partner on those sort of things.
Okay, thanks.
If I understood correctly, Monica also asked if you engage in such a project in Europe.
Oh, in Europe, yeah. I mean, Tetyana, correct me if I'm wrong, I don't think we've seen many in Europe, correct, yet?
Because actually now there is a project for the biomarker use for the stratifications of the patients, and basically what you're doing was exactly that.
Yeah.
I can say what facilities, what capacities, let's say, we have in Europe. So what we're doing in Poland and Ukraine is basically a collection of biospecimens according to criteria. In their very big network of hospitals. The same in Romania, Bulgaria, Croatia, Hungary, Macedonia. Hopefully, I not forget anybody. The next, let's say, level of services is a laboratory services. In this direction, we have big laboratory, blood services, and tissue also services in Bulgaria, where they process DTC, which were mentioned by Shawn, PBMC, BMMC, and all types of blood processing. In Ukraine, we have our licensed pathology laboratory, which also participate together with their clinical network sites in the diagnostic process of patients, providing full pathology services. And in Kassel, we have a tissue biomarker unit, which provides services for companies ordering for clinical trials. So I think, if I'm not mistaken, Shawn will give details. So our tissue biomarkers and IHC services in Kassel. So we can proudly say that this unit makes significant input in the development of several IHC markers. And HER2, it seems to me, it was one of their So that's why this is what we can do in Europe. The concept that today we decided to present, that nevertheless where our unit is located, in California, in Alabama, in Texas, in Sofia, even in Ukraine, with all difficulties that we know here today. So we are able to ensure their full annotation the workflow, life cycle for the product. So the question, if scientists exactly know what he wants, yeah.
Oh, Tatiana, I think it muted you for some reason.
Let's mute yourself.
No, it's okay. It's yeah. So we just need very detailed specification from the client, and we are ready to find the solution to shorten this long way from 12 years, 15 years to several months, sometimes even several weeks, if it is already on inventory. And I think this is a clever idea because for today, money and business are serving on the first come basis, yeah. So who will get the finish first will get all the stars.
But I understand that these assets you mentioned, they can be used, can be included in any kind of research or funded project as something which is used as a subcontracting in a form of subcontracting. You are not partnering in the discovery process.
No, this is a one window, because you go to the sales, to the discovery sales. You give all details, what do you want to do. Our sales and feasibility team create your personalized route to receive what you need. So this is you make let's say one window concept. So you make a contract with the Discovery Life Sciences in US, and this is already our task to organize the process in the way where by the end of the day you will receive exactly what you need. If we need to involve the other divisions, it's on our side, it's our service. We know what we can do and what kind of priorities we need to build in order to get the result. Sometimes we serve some orders in the frames of the intercompany processes. So we can collect in one country, after send to laboratory in another country. On the next stage, it will go to Germany. On the next stage, it will go to Alabama on sequencing. At the end of the day, you will receive what you want.
Okay, you are a great salesman.
Yeah,
I think everybody
I know. Thank you, I know.
In which way our life sciences can be used in any development? So that's very interesting. There is a if there are no other questions from our guests, maybe I would ask a question: what are the next steps in development of the company? What are strategic ambitions? Are you going to grow, to collect more samples, to target more targets, or maybe you are kind of directing into yet another type of services?
It's a great question. So I think there's a couple ways we're moving right now. So one is, the biobank. We do know a lot about it. It is not fully characterized yet. And so there's a big effort to get the biobank fully characterized. In terms of additional technologies, the one that we're most excited about is when we talk about single cell biology. So in the tumor that I showed, we had to dissociate that first, right? And so even though we saw our clusters of immune cells and tumor cells, we don't know where those were in the parent tissue, right? Were the immune cells on the outside? Were they on the inside? Because that's going to really predict how the compound or how the therapy would work. So what we're now leveraging is the ability to take a slide, an H&E slide from a site, and actually go ahead and understand all of the transcriptomics, so every gene expressed within that cell, but in the dimensionality of the parent tissue. So now we can know exactly where the cell is, what it's expressing, and who its partners are. And so this is spatial biology. It's a big focus of ours, both in multiple divisions. Our Castle division is focusing heavily on this from the immunofluorescence side, and our US facility in Huntsville is focusing very much on this in the genomic side. So understanding spatial biology now is becoming a huge focus.
Okay. I tried to unmute it with spacebar, but it didn't work this time. But how about, this is very interesting for our community also, how about artificial intelligence in your services?
Yes. Okay. So I forgot to mention that, which is amazing because our COO, as Tetyana can relate to, it loves artificial intelligence. He loves AI. And so this is a big part of what we're doing.
That's why I ask.
Yes, a huge part of what we're looking to get into, especially because we do have so much data now. And so AI is definitely on the forefront. We're talking about having it do quite a bit for us. A lot of additional, being able to look at the path reports, look at the slides, understand what's going on there. AI is a big focus of ours going forward.
Okay, so I understand this can be kind of a field for the discovery collaboration options with Discovery Life Sciences.
Yep.
That's great. We already exchanged some ideas how to make it happen. I mean, how to get closer to engage in dialogue. That's interesting. Yeah, there's a comment. There's a comment regarding this misunderstanding. I'm not sure if this is misunderstanding about the projects, EU projects, which I call EU projects because they don't necessarily mean the projects are realized in Europe only.
Yeah.
Because there are some kind of partnerships also with US partners, so that's okay. Any other questions from the public? This is not a question, I believe. At least not to us. Okay, that was very interesting. We have still a few minutes left. So I don't have a question because that was so specific. So I'm not specialist to go any more deeper. We are very much interested. I'm as mentioned before in organizing this personalized medicine. Right now we are trying to organize this on the Life Science Open Space platform when we still don't know exactly, for example, what is the difference. I mean, we know, but that's the decision if we should call it personalized medicine or precision medicine. Yes. On the platform, which is better to describe the issue.
Yep. Yeah, that's great point.
But do you know the answer?
I don't. I'm not gonna lie. I like precision medicine better because it does really show like what we're trying to do here is so specific. But yeah, it's funny. I was meeting with some folks in Chicago yesterday, the day before, and they were using precision medicine where we'd always use personalized medicine. So it's still out there, trying to decide which one. I like precision medicine, even though I just gave an entire talk on personalized medicine.
Probably it's depending from it maybe can be results from taking into consideration the perspective. If the scientist is looking, maybe he's talking about the precision medicine, and when the doctor is talking or the patient is talking, he's talking about the personalized medicine. But they are talking about the same actually. It's only the approach. Okay, thank you for that. I think I may now.
We have a page on the platform. Shawn has a profile on platform. So if you need any details regarding the today's presentation, so you are welcome to contact us on the platform. So we're always in your disposal for any kind of services which we're specialized for. And thank you very much again, Shawn, for this presentation. Probably this is the first case, yeah, during this year at least, that we have a presenter who present us at 3 o'clock in the night.
Yeah, exactly.
So, and for
Very
this,
early
in our
breakfast, yeah.
additional, yeah,
Actually, this is
appreciation.
supper for you.
Yeah, and hope to see you in Krakow. I already promised to compensate all these night presentations with a good team, something tasty, beautiful, and amazing in Krakow. You're welcome.
Yeah.
Awesome.
Are you going to San Diego to bio convention?
Yes, I will be there.
So we can meet there. So
Perfect.
Have a beer and to continue the talk. Maybe you will learn more. So fine, I'll find you.
Okay,
Somehow.
awesome.
Will you be on the Alabama stand or something like that, or participating in the conference?
Yeah, so I'll be on the posters. My poster is 1 to 5 on Tuesday.
Okay.
Yep.
Okay. So I'll find. Good to know. Thank you very much for this. A few announcements. Last minute announcement. Maybe this is important. This is first actually announcement. Still, I would say unofficial because we want to make it more official, but because the group is not so big, I can tell it. Yesterday we received information that we have succeeded. I mean the region, because we received 2 projects at the same time. One organized by the Marshall Office and second organized by us, or both in digital health. One is more focused on virtual reality and virtual reality technology applied to health, and second on digital health. In this, with these 2 projects, Małopolska region receives the title of Innovation Valley in digital health. So a lot of opportunities will come after that, and we will communicate about this and also what's come after that. What does it mean to receive this title? Because this is related to projects. So this is something interesting, and we'll communicate about this very soon. Maybe tomorrow, maybe the latest next week. Next week we meet with Kraków City. This will be also very interesting, a very interesting meeting, especially because the elections. I don't know what to expect. The next week meeting is live. It will be.
Sorry, Kazimierz, not next week. The second. Next week is
yeah, right. Next
holiday.
week is free. After Easter, the first week, first Thursday after Easter is live. We meet with Kraków in Klastra LifeScience. So if someone who would like to be present, please register. You will be asked to register. If someone wants to come from Alabama to take part in this breakfast, you're welcome. Let us know in front of before this event. Yes, we wish you still now the happy Easter, because this is the last time we meet before Easter. Shawn, good night. Have a night. Have a nice dreams.
Thanks.
Thank you.
To all others, good day.
Thanks.
Thank you very much.
Thank you.
Thank you,
Bye.
everybody.
Okay, goodbye. Have a nice day.